The next organelle is the vesicle. And again, I just want to draw your attention to the fact that we have a bilayer that you basically create a hollow ball. So you've got your bilayer, and then here's the hollow part inside, and you can basically carry anything inside of a vesicle. Now, vesicles are going to have different roles depending on what's inside of them. They're only small spherical sacs that are literally made of lipid bilayer, and they're all classified according to their contents. What are they carrying? They often move to and merge with the plasma membrane, releasing their contents, which I was just talking about in the previous video. So we have a vesicle here, which then starts to merge or fuse with the cell membrane, releasing the contents to the outside. It also goes the other way when you have a bunch of contents or a large particle that's going to be absorbed by the cell. The cell membrane starts to kind of cave in on one side, and it pinches off, and then what you get on the inside is a vesicle. So these are going to be examples of vesicles that are classified according to their contents. So I would also like to talk about the fact that some means vesicle, and lyse in this example, well, lyse just in general means to break. Like many videos ago, I talked about hydrolysis, and the lyse in that part meant to break. So a vesicle that breaks. This is a vesicle that contains enzymes. So this is a photographic image of a lysosome, and the dark purple area you see in the middle, that is, excuse me, enzyme. So why do you need organelles inside of a cell, which are basically just lipid bilayer balls full of enzymes? Easy. You needed to digest food. You also needed to digest old organelles. If you have a mitochondria that no longer functions, you need to be able to break it down and recycle the parts that it's made of. And also digesting other cells. Your white blood cells are going to have lots of lysosomes because their whole purpose is to destroy anything that doesn't belong. So the function of the lysosome is digestion. So that is an example of a lysosome which comes in contact with, in this case, it looks like a rod-shaped bacteria, and you've got the enzyme inside to break it down. This is just an example of some of the enzymes that can be found inside of a lysosome. You do not have to remember all of these, but I will focus your attention on the vocabulary part of this. So for phosphate esters, and again, you don't have to remember this, but I just want you to look at the name: acid phosphatase. Nucleases, DNase, RNase, protein digesting enzymes, collagenase, cathepsins, the in means protein, proteases, beta glucosidase, hexosaminidase, blah, 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 ACE. That's the point, ACE. That's what I wanted to say. I have a hard time pronouncing it as well. So the suffix ase means enzyme, and the suffix in means protein. Okay, now another function of lysosomes occurs during fetal development. So here I've got different snapshots of a fetus, the hand of a fetus during development. So the very first indication of a hand being formed in the uterus looks like a flipper. Basically, it looks like a flipper of like a dolphin or a whale or a seal. And now you can actually see the individual skeleton framework of the fingers being formed. Now, the thing here, 48 days after conception, or after the egg and the sperm fuse together, you can see the beginnings of fingers, but you're going to need enzymes to come in and kind of break down the skin, because obviously we don't have webbed fingers like a frog, right? So you're going to need enzymes to break down the skin between the fingers so that you end up with a normal looking hand and not like a freak frog hand. And so that's another function of lysosomes during development. Another kind of vesicle is peroxisome. Again, some meaning vesicle. In this example, peroxy is named for the hydrogen peroxide that these vesicles produce when they break down alcohol and they kill bacteria. They also break down fatty acids, which the mitochondria use as energy. Other vesicles, again, all named for the contents that they contain. We have glyoxysomes. These are specialized peroxisomes in the seeds of some plants, and they break down stored fats to provide energy for the plant embryo in the seed. So what I want you to think about is, what is a seed? A seed is a plant embryo. If we know what an embryo is, it's when the sperm and the egg come together. That's called an embryo. Now for plants, there are some plants that produce seeds. And what that is a plant egg and a plant sperm already fused together. And it's contained within this structure called a seed, where usually you have the embryo as a tiny little part. And then the rest of this is nutrients, because seeds can be dormant for a really long period of time. And they're dormant because it's basically waiting for the right environmental conditions in order for this embryo to sprout and to start to grow outside of the seed. So the oldest seeds, I think, that I've read so far that have been able to be germinated or to grow were lotus seeds that were 1,000 years old. So how does this embryo, or let's just call it like a pre-baby plant, it's a potential plant, right? It's a plant embryo. How does that stay alive for 1,000 years? Well, part of it is because you have all of this goodness, all of this nutrients inside the seed, and it's completely dependent on these glyoxysomes to break down the nourishment in that seed in order to feed it to the embryo so that it can stay alive and so that we can grow seeds after 1,000 years. A really good real world example of how to put this in your mind is popcorn. Okay, so one corn kernel, one of the little yellow kernels, that is a corn seed. There's actually an embryo in there. And when you expose that to heat, when you pop it, all of that puffy white goodness is the nourishment that was meant for the embryo. A very general term for any vesicle that is formed by the process that you see happening here of substances that are being taken into the cell is called an endosome. Again, some, vesicle, endo, inside. So anything going into the cell and forming a vesicle because it's going into the cell is an endosome. A very interesting vesicle that you find in unicellular organisms that live in fresh water is something called a contractile vacuole. This is a vesicle that can contract and get rid of excess water. So you see that up here and down here. These are two contractile vacuoles. So what's happening? This is an organism called a paramecium. It is a protist, like I talked about in a previous video. We have the 6 kingdoms, and protista is one of them, where you kind of throw all of the things that don't belong in plant, animal, fungus, eubacteria, or archaebacteria. Everything else goes into protista. These are unicellular organisms that live in fresh water. Now, what happens when you live in fresh water? Inside the cell, or sorry, outside the cell is 100% water. Fresh water is pure water, right? Inside the cell is less than 100% water. So what happens is that because there's a higher concentration of water outside the cell, water is going to tend to rush in to the cell. And if the paramecium does not have a contractile vacuole, it will literally blow up. Like if you fill a water balloon too full of water and it pops. The paramecium will explode because inside of the cell, you don't have 100% water, obviously, because you have like a nucleus, you have organelles, you have dissolved vitamins, you've got proteins and carbohydrates and all of these things. And the more things that you add to the inside of the cell, the lower the water content becomes. So if there's 100% water outside, less than 100% inside, water is going to go in. And so you need the contractile vacuole so that it doesn't blow up.